This QuantiGene training tutorial is designed to illustrate 3 key steps, which include setup of the capture plate and overnight incubation, washing and incubation of the capture plate with the preamplifier working solution, and reading of the capture plate in the luminometer. The sample type demonstrated in this tutorial is tissue homogenates. For day 1 of the assay, remove the capture plate from the 4-degree storage and allow the plate to equilibrate to room temperature for at least 30 minutes. In this order, add nuclease-free water to a 15 milliliter tube. Next, add lysis mixture, followed by blocking reagent. Vortex the QG probe set for 10 seconds. Quickly spin the tube and add to the 15 milliliter tube. Prepare a similar working probe solution for the housekeeping gene. After room temperature equilibration of the capture plate, open the sealed pouch and prepare the plate for addition of the working probe solutions and samples. Vortex and transfer the working probe solution from the gene of interest to a plastic reservoir and pipette 60 microliters of reagent into each well of the capture plate that will be assayed. Vortex and transfer the working probe solution from the housekeeping gene to a plastic reservoir and pipette 60 microliters of reagent into each well of the capture plate well that will be assayed. From your tissue homogenate source plate, transfer 40 microliters of sample to each well so that the total volume in the well is 100 microliters. For the background controls, use 40 microliters of homogenizing solution. To prevent evaporation, apply firm, even pressure across the seal using a soft rubber roller so that the numbers and letters of the plate are visible beneath the seal. Calibrate the oven using an NIST digital temperature meter from Affymetrix and ensure that the probe temperature reads 55 degrees. Place the plate into the incubator and incubate overnight for 16 to 20 hours. After incubation, carefully remove the foil seal and add 200 microliters of 1X wash buffer to each well. Quickly invert the assembly over sink and blot onto several layers of paper towels to remove any residual solution. Wash the plate a second time by pipetting 300 microliters of 1X wash buffer into each well using a multi-channel pipette. Quickly invert the assembly over sink and blot onto several layers of paper towels to remove any residual solution. Wash the plate a third and final time by pipetting 300 microliters of 1X wash buffer into each well. Quickly invert the assembly over sink and blot onto several layers of paper towels to remove any residual solution. To remove any remaining solution, invert the plate, place on paper towels and centrifuge at 240 G for 1 minute. After centrifugation, add the 2.0 preamplifier working solution to each well that is being assayed. After incubation, repeat the wash steps as previously described. Add amplifier working solution to each well and incubate at 55 degrees for 1 hour. After incubation, wash as previously described and add label probe working solution to each well. Adjust the temperature of the incubator to 50 degrees and incubate for 1 hour. After incubation, repeat the wash steps as previously described. Add the chemiluminescent substrate to each well and incubate for 5 minutes. Place the plate into the luminometer and set the integration to 0.2 seconds. For best results, read the plate within 15 minutes. This concludes today's video tutorial on the QuantiGene 2.0 assay.